Hello guys, today we're going to talk about the urinary system. Of course we need to know the function of the urinary system. The first and most important one is the regulation of water and ions. And I want you to think of this balance of water and ions as a whole entity. Why is that? Because wherever you have ions, you'll have the movement of water through, of course, osmotic pressure, one of the most important things. Second of all is that wherever you have ions, wherever you have water, there will be, of course, ions because of in order to balance the osmotic pressures again, or through diffusion or through different mechanisms that we already discussed in other videos. So the second one is the excretion of bioactive substances, detoxification in other words. Some drugs are being detoxified mainly by the liver. Of course, the major source and the major location of detoxification is the liver, but of course there are other drugs that work through the circle of the excretion by the kidneys. Third is the regulation of blood pressure. Now this, of course, is a very complex mechanism, and there are more than just one different mechanism in order to achieve this final result. And one of the most important systems you will discuss specifically in the kidney is the renin-angiotensin-aldosterone system that actually plays a role in the regulation of blood pressure. Of course, there are also a couple of more that we'll cover in details that work in order to achieve the same goal, and we'll discuss them, of course, thoroughly in just a bit. Next one is the erythropoietin production, which practically means that as a consequence it controls the amount of red blood cells present in the blood. And of course, again, this has to do with the proportion of cellular and non-cellular elements in the blood, and indirectly, of course, again, with the let's say the concentration of the blood. And then we have the activation of the prohormone vitamin D. Here it's important to understand that this is the location of activation, not necessarily not production. It's also interesting to notice that the vitamin D is considered to be a hormone, and this is because it actually exerts multiple functions, and there's a whole pathway when it comes to the function and the reactions to vitamin D. And the last one, of course, is the gluconeogenesis, which means production of new glycogen, or production of new glucose. And of course, this happens in cases of starvation and prolonged fasting periods, again, production of glucose in extreme cases. So let's talk a bit about the structure and the morphological, let's say, aspects of the kidney. So we have, this is the lobulated structure. There are specific renal lobes that are composed of two major sections, the cortex and the medulla. And of course, then later on, we're going to see the calyxes, which are the areas where we actually concentrate and receive the urine in the specific location. Of course, the function of the kidney is primarily urine production. Of course, the cortex and the medulla are different structures that contain a bit different, let's say, aspects of the same structures. And most importantly, most notably, we're going to see the nephron. Now, the nephron is the functional unit of the kidney. Before we move on to discuss more about the nephron, I just also want to mention that we have in the sides of the medulla, we're going to find the renal columns, which are the supportive structures, again filled with very important components such as blood, such as lymphatics, and we'll see specifically in the microscope in just a bit what exactly is contained within these structures. So let's talk a bit more about the nephron. So the nephron is composed of two different components, of course. There is the renal corpuscle and the renal tubule. Now we'll discuss first the renal corpuscle, its functions and its morphology, along with with specific unique features, and in the second phase we'll discuss about the renal tubule and again its function, morphology, and aspects. So there are two different types of nephrons. The important thing is that because in order to achieve this regulation of water and ions, and of course this detoxification, all these functions of the kidney that we discussed, we need to have a very, very rich vascularization. So this happens, of course, through the renal artery and renal vein. Now, as you can see in the schematic, there are a lot of subdivisions and spreadings, let's say, of the artery, from renal artery to segmental to interlobar, arcuate, interlobar, and afferent arterial. Now, we'll discuss a bit more about the afferent arterial after we discuss about the function of the glomerulus. So the point is that we have arterioles, afferent arterioles that bring in the blood, and efferent that practically takes it away. It is important to notice that the whole complex structure of the renal tubule, yes, and the renal corpuscle, in other words, the whole nephron, is a structure that is highly vascularized. And this is because we need two different functions in this case. Of course, the filtering as itself, the filtering of the substances and the ions and the water in the bloodstream, of course, but also we need to secrete we need the renal secretion, the renal filtration, and the renal reabsorption. Some molecules have to be excreted in the urine. Some molecules have to be, of course, reabsorbed, because we're going to see that the kidney, the nephron in the beginning, just by the initial filtration, is not a perfect procedure, and we have a lot of losses from water, ion, and other different types of saccharides and other smaller molecules. And we'll see exactly how this whole reabsorption and what type of molecules are being reabsorbed. So again, the logic of the nephron is to simply filter out a very big mass, a very big, let's say, a lot of components from the human blood to the filtrate, and then select which of them are going to be toxic waste and simply be passed on in the urine, and which of course are going to be reabsorbed in order to maintain the balance that we need to have in the plasma. So in order to achieve this, of course, high necessity for blood, for oxygen and other molecules, we need to have both the peritubular arteries and the vasa recta. Now these two are the specific, let's say, capillaries present and covered throughout the whole structure of the nephron. So there are two different types of nephrons based on location. There's of course the cortical one, which is present exactly within the cortex and the, let's say, high to mid levels of the cortex, and the lower ones that is more close to the medulla. For example, this one, this is the juxtamedullary nephron. Now the term juxta is actually means approximate, close to, so it's really easy to understand juxtaglomeruli, which is close to the medulla. So let's go and talk about specifically the renal corpuscle, and then move on to the tubules, and then discuss the functions of each parts. So the renal corpuscle, as you can see in the picture right now, there's a very complex structure that starts with the afferent arteriole, the artery that brings in the whole blood as we want it to be brought in. It passes on the blood through the very, very extensive network of the glomerular, of this glomerulus. Now all of these contents, you can see, is just the same arteriole that comes in and forms this very, very complex structure, as you can see in the graphic. Now the point is, why do we have that? Why exactly would we need this, let's say, these many twists and many turns? Well, because again we increase the surface area, because again we need a much bigger surface than just the simple continual, let's say, simple tube of the artery, in the capillary in this case, the arterial in this case, because we need to actually maximize the amount of filtration and amount of passage from the blood plasma to the area of the Bowman's capsule right here. So this is the logic of why we actually need to have this, let's say, this glomerulus present, this very, very extensive network of increasing the surface area of the arteriole. Now we're going to find different types of cells within this renal corpuscle. First off, we're going to find the juxtaglomerular cells. Now these are cells, they're smooth muscle cells. They have very unique functions, which will be discussed along with the macula densa. So just for a second, forget this part, and let's move on to the next central part of the glomerulus. Now the glomerulus is going to have these arterioles are going to be covered with three different cells, because here again we're going to find a barrier yet again, the glomerular barrier. So in this case, we're going to find the inner part is going to be again with the endothelial cell, as always. It's against an arteriole, so we're going to find again the ependothelial cell. Exactly beneath, we're going to find the basal membrane, and exactly above, we're going to find these unique cells called podocytes. Now, podocyte actually comes from the word podo, which means foot, because based on its morphology, it looks like it has spreaded legs throughout the whole, let's say, area of the arteriole. Now, the function of the podocyte is again to manage and to assist this filtration, to selectively allow specific molecules to go from the blood plasma to the Bowman's capsule, as you can see. Now, we're going to find also specifically that in the Bowman's capsule, we're going to find the visceral and the parietal layer. Of course, the visceral layer, we already discussed it. Again, the visceral layer is again the presence of the podocytes right here. This is the visceral layer of the Bowman's capsule, and the parietal layer is going to be covering out the whole segment and the outer surface area of the rest of the capsule. The point, of course, is here to actually have a specific area in which the filtrate will be present in the renal corpuscle. So this is the basic features of the renal corpuscle. We'll discuss again the mesangial cells, but because the mesangial cells, the juxtaglomerular cell and the macula densa play a different role specifically when it comes to filtration and to adjusting this filtrative capacities and properties of the kidney, will be discussed in just a bit. So we're going to have again the vascular pole and the tubular pole, because again this is the area where the vascular component is, the arterial, and the tubular is where the renal tubule starts to actually starts. So the important feature is again this trifecta, this endothelial basal membrane and podocyte, and exactly what is its role in this filtration. So let's check it out. So in the innermost part of the this arteriole we're going to have here, we're going to find this fenestrated pores of the glomerular endothelial cell. So this is again the endothelial surface, which is specifically highly fenestrated with many pores that allow of course the passage of contents, but most importantly it blocks the cells from the blood to pass through the filtrate in the Bowman's capsule. So this is the first barrier that we're going to find here, exactly right here. This is the pores, the fenestration of the endothelial cells that block out the cells, and of course very big other molecules to pass by. Exactly as always, beneath the endothelial cell we're going to have the basal membrane. Now the basal membrane here, also called basal lamina in this case, is actually the area in which we're going to find the second level of the second barrier in order to again control what passes from the blood to the filtrate in the Bowman's capsule. Now the basal lamina here prevents the filtration of large proteins, for example albumin, which is of course is important for the concentration, the osmotic pressure of the osmotic concentration of the blood. Now it's also important to notice that here in this basal lamina we're going to find glucosaminoglycans. And why do we care about that? Well, because these components of the ECM that are dispersed and deposed inside the basal lamina in this case have a negative charge, and as a consequence it actually plays one more role into the let's say the repulsion of controlled charged ions. So this is again the second line of defense that prevents again large proteins and like reduces the amount of ions that passes by due to this ionic charge of the glucosaminoglycans within this basal lamina. And the last one of course is the podocyte. Now the podocyte, the foot cell also called, now this is the cell that actually extends this whole processes, this whole cytoplasmic processes in the whole layer of this basal lamina. Now the sub parts, the smaller parts of the cytoplasmic processes are called, the pedicels. Now these pedicels, the term actually means little foot from Latin in this case. Podos from Greek and pedicels is from Latin, and the point of this structure of the pedicel is to cover up the whole section and control in two ways what actually passes by. First off is the direct touch, the direct, let's say, approach from the pedicel to the basal lamina that blocks out, of course, a significant amount of components. And the second one is it can be found within this filtration slit. Now, it is important to understand that within these spaces that are formed between the pedicels, we have different specific proteins that are actually that block out the passage of components from one point to the other. Now these are going to be called slit diaphragms, and the slit diaphragms contain specific proteins that are called nephrons. So this is a very, again, the same picture, but again from a cross section that actually allows you to visualize better this whole process. Again, we have the glomerular filter. The first layer is the fenestrated capillary that allows only small proteins to pass by, only few, that blocks out the cells and allows only small proteins to pass by along with ions and other components. And large proteins, as I told you, are being of course blocked partly by the fenestrated capillary epithelium, and most importantly and primarily from the basal membrane, which of course again contains the both ionic charge, negatively charged ionic components, the glucosinoglycans, and of course blocks out the small protein, blocks also parts of middle-sized proteins. And lastly, the big proteins, the smaller proteins are being blocked by these fenestrations, by these, let's say, the slit diaphragms that are present between the pedicels. Now this is the pedicel of the podocyte, and these are the fenestration slits where we have the spaces between the pedicels. And let's say connecting these pedicels, we're going to find these very, very thin lines that are being called the slit diaphragms, which of course contain proteins, one of which, and the most important of which, is nephrin. So this whole structure blocks out cells, proteins, a large amount of ions because of the charge, and of course other components such as vitamins, such as urea, such as other components, hormones, and so on and so on. Of course, it's really important to understand that again, this is the first stage of filtration, and of course it's incomplete because a large amount of ions passes by, and it's very, very important that we reabsorb many of these ions depending on the necessity of to balance out the blood pressure and necessity to balance out the ions. Again, it's important to understand that the ions, the component, the amount of the concentration of ions within the blood plasma indirectly controls the volume of the blood. So this is very important to understand. Now, this is again the same picture that the same picture we saw before, but this is the first example of the histological, macroscopical actually, morphology of the renal corpuscle. This is exactly again the same picture. This is the glomerulus. Now this is the parietal layer of the Bowman's capsule, and here we have the sinuses, the empty spaces, of course. And now these are the components we'll discuss in just a bit. And here we're going to find these the three types of cells: the endothelial cells, the mesangial cells. We'll discuss the mesangial cells in just a bit, and of course the podocytes as we know them already. So I just mentioned again the mesangial cells, and this is the perfect idea, the perfect moment to discuss what exactly is it that they do. Now these of course are supportive cells. Their location suggests it, and of course their function again also proves it. So these are supportive cells that have dual or maybe even triple roles. One is immune, which is subdivided in two rows: the phagocytosis of protein aggregates, and of course other small components. This is very important. This can actually be a very nice question when it comes to later on in pathology. You'll see pathologies of the kidney based on the function of mesangial cells, so keep them in mind. And as a consequence, of course, because they're immune cells, they are able to secrete cytokines and initiate inflammatory reactions and furthermore immune-related functions. And lastly, and very, very importantly, is the contractile function. Now, it is important to understand again that whenever we have a vessel, the blood pressure of the vessel is controlled with many ways. One of them is the diameter of it. Of course, the most important part is the diameter of the vessel. Now, this diameter can be altered and changed, where it discusses in the cardiovascular part, but this is a very nice opportunity to talk about it again, is of course the contractility of the smooth muscles. Whenever we have smooth muscles in the periphery, in the middle layer, the tunica media of the capillaries or vessels or whatever vessel it is, and we have the activation, the constriction of the smooth muscles, we have the decreased diameter, and as a consequence, we have the increased pressure. So this is a mesangial cell. Based on these contractions, they alter the local pressure of the glomerulus within the content and within the lumen, let's say, of the glomerulus. So these are the basic parts I want to discuss about the renal corpuscle. Again, we should proceed to the proximal, the tubules, the renal tubules. We're going to find four basic structures: the proximal convoluted tubule, also called PCT, the Henle's loop, which has actually specific segments, the thin descending, thin ascending, and thick ascending, so descending, ascending, ascending. And then we have the distal convoluted tubule, which also is abbreviated as DCT, and lastly the collecting duct. Now, what exactly is it that these ducts do? Before we discuss the function, it's ideal to understand how they look like and where they're located and exactly what's the rationale. Of course, the whole rationale of the renal tubule is to reabsorb molecules and contents. So of course we're going to find cells that of course have again microvilli, because of course the ideal function of microvilli is the reabsorption of whatever it is, of whatever molecule we have within the content. And in this part of the renal tubule, we're going to find cells that have tall macrovilli, very dense, and others are going to be shorter or sparse. So the point is that the macrovilli, we're not going to be necessarily tools that are going to help us that much to differentiate between the different types of tubules, but this is again one of the components that we should discuss. So first up, let's realize and let's really understand what we're going to find in the cortex and what we're going to find in the medulla, which tubule can be located where. So here we're going to see that this section of the cortex, again, keep in mind that always the renal corpuscle should be within the cortex, either close to the medulla, the juxtamedullary ones, or the cortical ones, and proportionally only 1 out of 7, if I'm not wrong, are present within the juxtaglomeruli. So most of the renal corpuscles, most of the glomeruli are going to be present in the cortex. So if you have in your head the section of the cortex along with of course the glomerulus, you should understand that you are going to find only the proximal convoluted tubule, which is of course, as the name suggests, is close to the to the renal corpuscle, because this is the first segment. And of course, one more segment is going to be the distal convoluted tubule that passes again by closer again to the corpuscle, the renal corpuscle. Now, the term that you saw macula densa before, macula densa in fact is a part of the distal convoluted tubule. That's why I didn't discuss it before, and I'm waiting for this segment to actually discuss it. So we're going to find again in the cortex only these two parts, these tubules, the proximal convoluted tubule and the distal convoluted tubule. And sometimes, because of its length and because of its whole very big projection we're going to find the collecting duct, which practically more or less extends from the cortex to the medulla. So we're going to find these two and let's say half of the collecting tubule. In the medulla, we're going to find only the segments of Henle's loop, which are of course of the lower segment. And in fact, the renal tubules is a very long structure. It actually extends from the renal cortex, where we have the corpuscle, to the very bottom of the medulla, the renal medulla. So in order to identify the different types of tubules that we're going to have present in the medulla or the cortex, we need to understand that the medulla is going to contain, of course, the Henle's loop and the collecting ducts. Now, this is a very nice actual slide that is going to help you a lot to identify which is what, and again, it restates the locations of the tubules in the cortex, the proximal and the distal again, and the medulla contains only the Henle's loop and the collecting tubule. This is a very nice slide. I really suggest that you use this slide to study with and to actually compare your visual representation slides from the histology guide and of course this slide right here.